doubts until dialysis? Any doubts to anyone until dialysis? Just 33. Where are others? Let me take that in yesterday. So many are not there for the class. What is the reason? Busy with the December celebrations? Anyway, so tell me, any doubts with respect to dialysis that we studied? Okay. Have we studied the countercurrent mechanism? Have we studied the countercurrent mechanism? No. So we'll be starting with the countercurrent mechanism. So what is countercurrent mechanism? Write down. What is the aim behind converting the ammonia to urea? What is the aim behind converting ammonia to urea? To conserve water, right? And here the countercurrent mechanism is also to conserve water, that is to so that the water that is removed from the urine is less. The most of the waste is removed, but not water is removed. So countercurrent mechanism is an ability to produce concentrated urine. What do you understand by concentrated? Hypertonic urine. Okay. You know loop of Henle, you know Vasodector. This both plays a role in countercurrent mechanism, and the blood and the flow of the filtrate in both in the loop of Henle and the Vasodector is in opposite direction. So if you see this diagram, can you see the diagram here? In the Vasodector, the flow is like this. Can you all see the diagram that represent a Vasodector and the countercurrent?
Yes.
Yes. Okay. So this flow, the flow is like this in the Vasodector, whereas the flow is like this in loop of Henle. So the counter means opposite. The flow of the filtrate is in opposite direction. Okay. Next, the medullary interstitium fluid. Now tell me, where is a nephron housed? Where is a nephron housed? In which part? Where is nephron present in kidney?
Ma'am, between medulla and cortex.
Yeah, but where? In the renal pyramid, right? It's housed in the renal pyramid, right? Come on. Yes, Rishikesh. It's in the renal pyramid. Now, this nephron which is present mostly the cortex में क्या है? Corpus Bowman's capsule, PCT, DCT. Whereas in medulla, there is, what is in medulla? Loop of Henle and collecting duct. Am I right? The tissue present in the medullary is called medullary interstitial fluid. The tissue that is present is medullary interstitial tissue, and the fluid is medullary interstitial fluid, which is present in where? In medulla. This tissue is in the range of 300 to 1200 milliosmol per liter. मतलब the range कितना है उसका? Gradient. It shows a gradient about 300 to 1200 milliosmol per liter. This gradient, a gradient, who is causing this gradient? NaCl and urea is causing the gradient. Human kidney can produce 4 times concentrated urine than the initial filtrate. So the filtrate which is formed in the Bowman's capsule and the urine that is coming from the collecting duct, what is the difference from here, this filtrate to this urine? 4 times more concentrated. Are you all getting the point? Please write down. Any doubts to anybody there?
Ma'am, ma'am,
Huh, tell.
ma'am, should we write this?
Can you see that mechanism of filtration concentration? Please write that. Slide, can you see that slide? Rishikesh, I'll get back to you about the opposite flow mechanism. When we study the mechanism, you will understand it more clearly. Okay, Rishikesh? Are you done writing this part? Mechanism of countercurrent concentration of filtrate by countercurrent mechanism.
Wait a minute, ma'am.
Okay. Write it down and do let me know once you're done. Okay? Yeah. Done?
That
Okay. Next.
Ma'am,
Now,
ma'am, ma'am,
now. Okay,
ma'am,
fine.
can you
Write down,
please
write
wait?
down. Yeah, I'll wait, but you should let me know because I cannot see you, right? You should let me know. That's the reason I ask you. Are you done?
Ma'am, just go to that slide. I'll take a screenshot.
Which slide?
Counter current mechanism.
Now you can see the slide. I'm showing you that slide only. Can you see the slide?
ma'am, you can see the slide of mechanism of urine formation.
Yes,
Mechanism
ma'am.
of urine formation. No. Can you see this slide now? Mechanism of concentration of filtrate.
No, ma'am.
No, ma'am.
Now.
Yeah, ma'am. Now. Yeah, now I can see. Done, ma'am.
Done. Okay. Now listen. Counter current mechanism. Here, okay, I'll explain you. Can you see the diagram of counter current mechanism? Pencil diagram.
Yes, ma'am.
Please draw this. Please draw this. I'll explain you. I'll explain the mechanism. Please draw this. Done. Draw this. I'll make you understand. I'll explain you the diagram.
Ma'am, we should label all these parts again.
Yes. Please label. The way it is, please label everything. When I explain you, it will be more easier for you. Done? Then my screen is not visible. Is the screen not visible to anyone?
It is visible, ma'am.
Okay, fine. Yeah. Who is this? Who is speaking now? Who responded to me just now?
Excuse me, ma'am. What
Yeah,
is
tell me.
hypertonic? What is it?
Hypertonic means high concentrated, where the solute is more, water is less, solvent is less. You're taking 10 ml of water. In that, you're adding not 10 gram, but any 3-4 grams of more of sugar. Getting the solute is more, the solvent is less. Hypertonic, water is less. Getting the point?
Yes,
Something
ma'am.
like that. Yeah.
Excuse me, ma'am.
Yes.
What have you written that isoto?
Isotonic, hypertonic. Can I explain it now? My dear students, can I explain it now? Are you all done writing? Okay, no problem. Write down. Yes, done. How long? Okay. Now everybody please listen. You know what is PCT, you know what is DCT, you know what is loop of Henle, you know what is Vasodector, and you know collecting duct. Do you know all these parts of nephron? Do you know all the parts of nephron? Can you hear me? Do you know these parts of nephron? Am I sounding very lately, or are you able to hear me? Then Sakshi, tell me. Then why you people do not respond? Quick, I need a quick response from you all. You understand? Everybody will be listening to what I'm saying. Now, see, the filtrate is formed here, right? This is a filtrate. The filtrate is moving from the PCT, which is PCT. Now, this is the medullary interstitium. Medullary interstitium maintains the salt. That salt is from 300 to 1200 milliosmol per liter. That salt is from 300 to 1200 milliosmol per liter. The gradient. Who maintained it? NaCl and urea maintains the medullary interstitium of medulla. Now, when Rishikesh, please listen. When the liquid filtrate is moving, here it is isotonic. Iso means similar to outside. The tonicity of the liquid is similar to outside here. Okay. Now, as the liquid is moving downwards, when it is moving and when the salt is higher in the lower concentration, what happened? The more of water comes out. When the more of water comes out from the filtrate, what happened to the urine or the filtrate that is here? It become hypertonic. Do you agree with me? We are just talking about the descending loop of Henle. When the filtrate is passing through the descending loop of Henle, the water comes out as it moves towards the 1200 milliosmol per liter. The urine or the filtrate will become hypertonic because there is less of water in that filtrate and more of waste. Do you agree? Now, when the same filtrate is moving in opposite direction upward with the ascending limb, when the same filtrate is moving upward, it will start losing NaCl. And when it start losing NaCl, what will happen? This will be hypotonic. The urine will become again hypotonic. Losing NaCl now, losing water, so it became hypertonic. But here, from the water, you're removing the NaCl. What will happen? It will become more hypotonic. As the DCT, it moves again hypotonic or isotonic. But when it moves to the collecting duct, what should be the urine now? What should move out when it's moving through the collecting duct? It is going from 300 to 1200 milliosmol per liter. So what should happen?
Water should move.
water should move out. Very good. It's because the more water will move out. When more water will move out, then now the filtrate that is present in the collecting duct, or the urine that is present in the collecting duct, will be more hypertonic. Are you all getting the point? Yes. Any doubts to anybody there?
Excuse me, ma'am.
Mm-hmm. Tell.
Why is the water moving out?
Because the salt concentration out is more. Salt concentration is more outside. The gradient is maintained from 300 to milliosmol per liter. At lower concentration, the gradient is 1200 milliosmol per liter. The salt concentration is more at the lower level. So the water will move from higher to from the filtrate to outside, because the concentration of salt is outside more.
Understood, ma'am.
So when the water moves out, the urine will become hypertonic. Right? Do you agree? Got the point? Now, this is how urine is getting hypertonic. Now, then this collecting ducts, many collecting ducts joins the common duct, that is called what? Many collecting duct joins the common duct. Which is that? Duct of. Many collecting ducts joins the common duct. Duct of Bellini, not Bertini. Duct of Bellini. Do you agree? Now, the urine that is in the duct of Bellini is hypertonic. This is how you get hypertonic urine. Now, what is the role of Vasodector here? I'm using a different color. The role of Vasodector is, from the collecting duct, some urea is also getting absorbed. This urea is taken by the Vasodector, and then it is moving in opposite direction and give it to the outside to maintain the medullary interstitium. Same way, the NaCl which is removed on the ascending loop of Henle is taken by the descending Vasodector. It is taken and then given out at the ascending Vasodector to maintain the medullary interstitium. So the flow in Vasodector and the loop of Henle is in opposite direction. That's the reason it is said to be a counter current mechanism. Did you understood how the urine is becoming an hypertonic urine? Got the point? Any difficulties to anybody there? Right, yes, listen to me. Yes, please concentrate. Is anybody else who want me to explain again? Yes, when I'm speaking, look into the diagram. Okay. The filtrate that is formed in the Bowman's capsule is moving in PCT. In PCT, the tonicity outside in the cortex and the filtrate is same. So it is said to be isotonic. As the filtrate is moving downwards through descending loop of Henle, the tonicity increases outside from 300 to 1200 milliosmol per liter. As the filtrate is moving down and down, more water is removed. Why? Because the salt is more outside, the tonicity is more outside, so the water is removed into the medullary interstitium. So when the water is removed from the filtrate, the urine will become hypertonic. As it get into a ascending loop of Henle, it is again going in a backflow, like from 1200 milliosmol to 300 milliosmol. As it going upward in the ascending limb, NaCl is removed, then the water or the filtrate will become hypotonic. Has it moved to the DCT, again it is isotonic. When it again come into collecting duct, it is going to the gradient of 300 to 1200 milliosmol liter. When it's going from 300 to 1200 milliosmol liter, as it moves further in the collecting duct, more and more water is removed, because at a 1200 milliosmol liter, more salt is there. It removes the more of water, and now the urine that is in the collecting duct is hypertonic, and this urine joins the go through the duct of Bellini and then into the renal pelvis. This we have studied. This is how the urine is formed, which is hypertonic. What is the role of Vasodector there? The role of Vasodector is, listen, what is the role of Vasodector? The NaCl, which is removed from the ascending loop of Henle, is taken by descending Vasodector and given out by ascending Vasodector to maintain the medullary interstitium osmolarity. Who maintains the medullary interstitium osmolarity from 300 to 1200 is NaCl and urea. NaCl is coming from ascending loop of Henle, whereas urea is small or bit amount of urea is from collecting duct. This too contributes to maintain the medullary interstitium or the gradual. Any doubts? Repeat what? Ma'am, please repeat. Last point. Amod, listen. What is the role of Vasodector? Whatever the NaCl is coming from ascending loop of Henle is taken by descending Vasodector and given out by ascending Vasodector to maintain the osmolarity. Who maintains the osmolarity of the medullary interstitium is NaCl and urea. Got the point, Amod?
Yes, ma'am.
Ma'am, can you please explain what you just said?
I said the NaCl and urea plays a role in maintaining the medullary interstitium. I have given this point in the notes also. The gradient is caused by NaCl and urea. You have written this point. The gradient is caused by NaCl and urea. Everyone. No, write down this. Can you see this? A small amount of urea is diffused out of the collecting duct and add up to medullary interstitium. The presence of such interstitial gradient help in an easy passage of water. As the filtrate flows down towards the collecting tube, more and more water comes out by osmosis, which makes filtrate hypertonic. Please write down these 2 points quickly. And do let me know if you have any doubts.
Done, ma'am.
Okay. Did you all understood now? Did you all understood? Vaibhavi, now next write down ornithine cycle or Krebs cycle or urea cycle. Please write down ornithine cycle, Krebs cycle or urea cycle. Okay. Now this we you know that ammonia is converted to urea to so that the water usage is less. So where does it happen? Happens in liver through a cycle called ornithine cycle, urea cycle, or Krebs cycle. Okay. The two molecules of ammonia combines with one molecule of carbon dioxide that converts into urea with the usage of three ATP molecules. What does it say? Two ammonia molecules combines with one carbon dioxide, and how many ATP is used? I told you energy is used. Water is safe. So three ATP is used to convert into what? Into urea. Are you getting the point? Now, maximum urea is found in hepatic vein and minimum urea is found in renal vein. Why do you think the maximum is found in hepatic vein? Why? Very good. As urea is formed in the liver cells. Hepatic vein is a liver connected to the liver. Since urea is formed in the liver, it is said to be the more is found in hepatic vein. What is the condition when the urea is more in the blood called? What is the condition? Very good. Uremia. Very nice. Now tell me what is the condition? No, uremia. Very good. Minimum urea is found in renal vein. Why? Why the minimum is found in renal vein? Because it passes to the kidney. Are you all getting the point? Please write down. Very smart. Done? Yes, any doubts? Anybody, any doubts? Anyone, any doubts? Okay, next. Hmm. Now, we'll be studying the Osmo Regulator or Osmo Conformer. Osmo Regulator means the animals which maintain the osmolarity of the body fluid irrespective of our surrounding media. So, for example, if this is a one animal, for example, fish, I'm drawing a fish here. Osmolarity is 100 inside the body, outside the body is 150. So what does this do? Animals which maintain the osmolarity body, whatever the temperature, whatever the osmolarity is outside, but they maintain it 100. If it is 200 outside, if it's 500 outside, whatever it is, but they maintain 200 itself. Those are called osmoregulators. Examples are all vertebrates, fishes, amphibians, reptiles, apes, mammals, except hagfish. Remember hagfish we studied? Most of the freshwater invertebrates. These all are osmoregulators. Got the point? Got the point? Sakshi, any doubts? Others, any doubts? Rishikesh?
Ma'am, freshwater invertebrates are exception or are they examples?
Which one? Exception is only hagfish. Most of freshwater vertebrates is not an exception, sorry. It is included into this. Okay. Exception is only hagfish in vertebrates. Done, everyone. Next, what is osmoconformers? Osmoconformers are they whatever if for example if this is hagfish or any kind of fish marine thing. So if it is animal is having 100 outside, and if it's outside 100 milliosmol in the inside the body, if the outside is 200, what does this animal do? It changes itself to the outside osmolarity, so it changes to 200. So they change the body fluid osmolarity with change in the osmolarity of the surrounding medium. Example, hagfish, all marine invertebrates, some of the fresh vertebrates. Very, very important is hagfish example for MCQ. Done. Now, how the kidney maintains the osmolarity or the osmoregularity of the role of the kidney. So the first thing is whenever the body fluid volume is increased, then what have the first condition we are talking about? When the body fluid volume increases, there is an increase in blood pressure, and this can happen due to excessive water intake. Also, the body fluid might get increased because of the improper functioning of the kidney, right? So the aim of the is to produce. What should be the aim when the water, अभी जब body में water volume ज़्यादा हो जाए, तो what should be the aim? To produce hypertonic urine. Hypertonic में you conserve more water. So or to produce hypotonic urine in this condition when the blood volume is more, when the blood pressure has increased. Come on, tell me.
Ma'am, could you scroll up a little?
Yeah. Did you understood what I'm asking? When the body fluid or the volume is increased, there's increase in blood pressure. So what should a person? Should the person need to throw the lots of water out of the body or to preserve or conserve the water? Come on, tell me. The aim should be to produce hypotonic urine. How? One is by increasing the ultrafiltration. When the volume is increased, there is increase in blood pressure, which leads to increase in glomerular hydrostatic pressure, and that leads to increase in glomerular filtration rate, and more filtrate is formed, and then there is hypotonic. When the blood, the second is by decreasing the water reabsorption. How? When there is an increase in blood volume, this activate the osmoreceptors, which will decrease the hypothalamus functions. When hypothalamus function is decreased, the release of ADH is also decreased. And when ADH is not released much, ADH usually absorb water. So there is a decrease in reabsorption of water that leads to hypotonic urine. Please write down this. And do let me know if there is doubt. The two-way where you can throw water when the blood volume is high, and then you can maintain the blood volume by producing hypotonic urine. Rishikesh, clear? Sanjana? Sakshi? Sakshi, listen. Are you listening, Sakshi? See. What happened now? 2 minutes. Sakshi, are you listening? It's time up. 1:30. When the body, in the body, when the water amount is increased, we need to produce or we need to maintain the book. Actually, the kidney maintains the body volume. So, if the body volume or the blood volume is more, the water in the blood is more, the kidney needs to remove the water. How does it do? It do it by two proper function. One is by increasing the ultrafiltration and other is by decreasing the water reabsorption. How by increasing the ultrafiltration? The urine, the nephron, whenever the blood reaches to the nephron, it try to remove a more filtrate. When it removes a more filtrate, then when it can work more, then it can remove more water outside. That is, when there's an increase in blood volume, there's an increase in blood pressure. That increase in blood pressure lead to increase in glomerular hydrostatic pressure, which increase in glomerular filtration rate. The rate of filtration is more and more filtrate is formed, and that leads to a hypotonic urine. So you're removing more of water there. The second method is by decreasing the water reabsorption. Water kidney में water reabsorb होता है किधर? Under the influence of hormone ADH. Whenever there's a more ADH is produced at the DCT, there's a more water reabsorption. So if you can decrease the water or ADH release, then what will happen? There is no reabsorption. If the water is not reabsorbed, it is thrown away from the body. This is how we can maintain the body blood volume. Did you got the point, Sakshi? Next. Next is when the volume of blood decreases. Abhi, first we did when the volume of blood increases. Now we'll be studying the volume of blood decrease hota hai, tab kya hota hai. When the volume of blood decreases, the decrease in blood pressure, then there's a decrease in blood pressure. The volume might decrease due to diarrhea, vomiting, the blood loss, excessive blood loss, dehydration, anything. That the volume of the body decreases and the water content is water is decreased in the body. Then we need to produce hypertonic. Hypertonic क्यों? हमें water को conserve करना है. We need to increase the reabsorption. So now the mechanism will be opposite. How? One is by decreasing the water, by decreasing the ultrafiltration. Okay, there it was increasing because we need to produce hypertonic urine. Here we need to preserve the water, so we produce hypertonic. So it is in opposite direction. By decreasing the ultrafiltration, what will happen? There is a constriction of the blood vessels, that is called myogenic control. That time when there is a constriction, the blood flow is less, so less filtrate is formed and less hypertonic urine is formed. Next is by increasing the water reabsorption, means more of ADH is produced, that will release the more of reabsorption of water, and that release to, when there is a more of ADH release, ultimately there is a more of reabsorption of water. जैसे ही water reabsorb हो जाएगा, urine will become hypertonic. So we are getting the normal blood volume again if the volume is less. It's exactly opposite to the first. Please write down and do let me know if you have doubts. Done? Please write down and do ask
No,
me
ma'am.
if you have doubts. Okay. Do it and let me know if you have doubts. Done? Understood everybody when you're writing? Are you understanding? Is there any difficulties there? Come on, tell me. Have you understood? Others? What about others? Each and everybody in the class. Have you understood? Okay. Next. There is one more method. There is one more method which where you can activate the that method is called RAS. Let us study about the RAS, which is a very important method. Okay. RAS. What happened in the RAS? Listen to me. First you listen, then I'll give you time to write. RAS is a third way where you can increase the water, okay, or you can preserve the water content. So how? Let me show it to you. Just a minute. Okay. So if this is a efferent arteriole divides into capillaries, right? In the efferent arteriole, there is a juxtaglomerular cells. In the efferent arteriole, there is juxtaglomerular cells. Together, it is called juxtaglomerular apparatus. The RAS stand for renin, angiotensin. This renin is different from the renin which is studied in digestion and absorption. That renin is double N. Here it is single N. So RAS is a renin, angiotensin, aldosterone system. This juxtamedullary glomerular apparatus contains special type of cells called juxtaglomerular cells. These are modified cells present in the efferent arteriole which stores inactive renin. It stores what? Renin, which is inactive. Are you understanding? Now, RAS gets activated when? When is the RAS getting activated? When there is a decrease in blood volume, decrease in blood pressure, decrease in glomerular filtration rate. Whenever the filtration rate is decreased, what happened? This cells, GC cells, detect the changes that the blood jowara, it's decreased in volume. The pressure is less. That's why this GC cells which contain renin, they release the renin from there. This renin enter into the blood. Once the renin enter into the blood, what will happen? Angiotensinogen, which is formed in the liver, gets converted into angiotensin 1. This angiotensinogen is a plasma protein which is synthesized in liver and it is made up of 453 amino acid. Important question. This angiotensinogen gets converted in the presence of renin to angiotensin 1, which is a 10 amino acid, that is decapeptide. This angiotensin 1 converts into angiotensin 2 hormone, which is 8 amino acid in the presence of enzyme angiotensin converting enzyme from alveoli. Clear? Now, this angiotensin 2 perform three way to conserve the water. First way is, what does it do? It stimulates the adrenal gland. In the adrenal gland, if you remember, there's a release of aldosterone hormone. So when aldosterone hormone is released, there's a more reabsorption of sodiums. When there's a more reabsorption of sodium, there's a more water reabsorption from the DCT. So you are getting hypertonic urine, thus you are preserving the water. The next is, what does it do? It will act on the smooth muscle and bring about the contraction of vessels, blood vessels. It brings about the vessels constrictions that leads to the normal blood pressure. The third is, in the hypothalamus, there are centers for thirst and thirst. So it stimulates the thirst center so that we can take more and more water to get our water back. So this is how the RAS plays a role in maintaining the or getting back the normal blood volume when the blood volume is less. Is it point clear? See, you can do one thing, write down. If you have doubts, I'll explain you again. First, write down, understand, and then write. The third way is by activation of RAS. Very, very important. Very important. Write down this much. Come on, quickly. Please understand and then write. I want each and everybody to understand what you're writing. Okay, next, write down. Please write down. Any doubts? Is it clear to all, each and everyone? I can see only Rishikesh responding. The question is to angiotensin 1. Okay. The question is to each and everybody in the class. Are you all understanding? Done? Madhura, any doubts? Okay, Srisi, no problem. I'll wait. But are you understanding? That's more important. If okay, right now. Done, everyone? Reshma, is it clear? Divyanka, Annam, Malachi, Sneha, Isha, Ishan, any doubts? Dia, Arya, Sania, any doubts? Come on, tell me quickly. Okay, fine. Okay, then I hope you all are done. If you have doubts, do let me know. So we'll wind up the class for today. See you all in the next class. Until then, keep learning and bye bye. Take care.